Hi, we are students from Harmony School of Advancement, and in this video, we will be explaining the ELISA test.
ELISA is based on the principle of antigen-antibody interaction. An antigen from the sample binds to antibodies attached to a solid surface. Then an enzyme-linked antibody specific to the antigen is added. The enzyme linked to the antibody reacts with a substrate to produce a detectable signal, usually a color change, which is proportional to the amount of antigen in the sample. The materials we use to conduct the ELISA experiment are microstrip wells coating antigen or antibodies, a blocking agent, a primary antibody, a secondary antibody, an enzyme substrate, and a washing buffer.
To conduct the ELISA test, first coat the microtiter plate welds with a specific antigen or antibody. Then add a blocking solution to prevent nonspecific binding sites. Next, incubate to block for a defined period. Then dilute and apply samples containing the target molecule to the coated wells. Next, perform multiple washes with a washing buffer to remove unbound substances and add primary antibodies to the specific target. Then incubate to allow binding. After that, wash to remove any excess primary antibody that was applied. Then incubate to allow binding to the primary antibody. After that, wash to remove excess secondary antibody enzyme substrate. Then add the substrate for the enzyme linked to the secondary antibody. Then incubate for a specific duration, and finally introduce a stopping solution to halt the enzymatic reaction.
In our analysis, the color intensity decreases from well 1 to well 12, which is consistent with decreasing concentrations of the antigen in the serial dilution. This part of the plate can be used to generate a standard curve if the initial concentrations are known, which is fundamental for our quantitative analysis. In 13 to 15, our positive control had a strong blue color, indicating a positive reaction and that our assay is working correctly. Controls are essential to validate the results of the assay. Wells 16 to 18 are negative control or colorless, which indicates the absence of the target antigen. For patient 1, wells 19 to 21, there is color change in the wells suggesting the presence of the antigen. For patient 2, 22 to 24, the reaction appears to be colorless, which indicates an absence of the target antigen.
For our English component, we documented our journey into truly understanding the ELISA test. During the startup phases of the experiment, we set up the basics of our Google site. Afterward, we researched this topic to understand the importance of the ELISA experiment and what the test is trying to accomplish. We then carefully conducted our experiment, making sure that we followed all of the instructions while documenting the results in our Google site. Lastly, we analyzed the data we collected and began composing a simple but concise analysis with a conclusion that highlighted all the important details we studied. These results were then published on our Google site for others to view and gain their understanding of ELISA testing.
For our social studies component, we described the historical background and connections of the ELISA test. The enzyme-linked immunosorbent assay test was developed in the early 1970s by a Swedish biochemist and immunologist, Dr. Eva Envall, and American immunologist, Dr. Peter Perlman. They published their work on the ELISA test in 1971, describing their method as a sensitive and specific way to detect and quantify proteins.
Thanks for watching Harmony School of Advancement.